Hello, everyone, and welcome to the Parkinson's Foundation Expert Briefing. I'm Dr. James Beck, Chief Scientific Officer of the Parkinson's Foundation, and it's a pleasure to have you here today. In today's briefing, we will explore the latest discoveries, where the science is headed, which I think is going to be really exciting today, and how these advances may guide new and more effective treatments. Before we begin, I'd like to just give a warm, generous thanks to Genentech Therapeutics for sponsoring today's expert briefing. Their generous support helps make programs like this possible, and we appreciate their commitment to the Parkinson's Foundation's mission. And as we go to our next slide, just want to ask everyone who's out there listening today, please tell us what your connection is to Parkinson's disease. This is something we like to do on a regular basis. A little poll just would pop up. And if you're on Facebook, just put into the comments what your connection is to Parkinson's disease. Just like to keep track of who our audience is, and our speaker gets to see this as well, and can really make certain we're recognizing our audience today, even though we can't see you. Ensure that what is being delivered is something that you find really interesting, as I think we will today. And so we'll just give that another few seconds and see where that goes. Great. Not surprisingly, a lot of people with Parkinson's or closely related to Parkinson's, a few scientists and physicians mixed in, healthcare professionals. Welcome, everyone. And really excited to move on with our expert briefing today. But before I do, I always like to just let everyone and remind everyone that our focus at the Parkinson's Foundation is to really make lives better for those living with Parkinson's disease. We enhance care and advance research. And these efforts are deeply rooted in collaboration with the Parkinson's community, ensuring that everything we do aligns with the needs and priorities of those who live with PD, you, and your loved ones. And today's program is just yet another example of how we're working with you to meet those goals. And one of our key initiatives at the Parkinson's Foundation is PD Generation. Here at PD Generation, we offer free genetic testing and counseling to people living with Parkinson's. And by participating, you can learn more about your genetic connection to PD and contribute to Parkinson's research. With one day that this information will help drive treatments towards a cure. And with your commitment, I'm sure we will. So I think this is an example of where together we can really make a difference in Parkinson's disease. So as we do every time, we're recording this expert briefing, and we'll make it available to everyone after this is over. And so if you have to step away and you miss something, or you have friends who weren't able to attend, we'll be able to download this and be able to watch it at your leisure over and over again if you like. I think this will be one of those oft-repeated ones. And as we talk about this expert briefing, let me just introduce our speaker today, Dr. Laurie Sanders. She's an associate professor of neurology at Duke University and a leader in translational neuroscience focused on Parkinson's and other neurodegenerative diseases. She serves as the faculty lead at the Duke Clinical Research Institute, where she also co-directs clinical trials and has led multiple series as a principal investigator. Dr. Sanders is widely recognized for innovative work in biomarkers and therapeutics, and with several awards and patents that are there to advance the field in Parkinson's disease. Dr. Sanders, thank you very much for your time and for joining us today.
Thank you so much, Jim. I'm gonna now see if I can share my presentation. Thank you, everybody, as I navigate through here. Can you see it okay?
Not yet.
Oh, okay. Let's see.
There we go. Starting now.
There we go. Okay. Thank you, everybody, for your patience as we get technology to catch up here. Thank you for this opportunity from the Parkinson's Foundation to speak with all of you today. I must admit that this is really such a privilege for me to be able to engage with all of you. And I will also say that for those of you that live in the South, as I do in Durham, you know that it's pollen season. So excuse me as I'm a little congested with the allergies right now, because it's a major pollen season for us. So thank you for your patience. So I did want to just mention a few of my disclosures, which again don't have any influence in sort of or any impact in what I'm going to talk about today, but just did want to mention that I do sit on a scientific advisory board for Neolea Bio. I will be talking a little bit about how we've used our patent for a blood-based biomarker today. And I will share a little bit about some of the studies that sort of integrate into sort of some of the very, very exciting work that's going around the world for a couple clinical trials and a new consortium that I'm so privileged to lead in terms of the gut-brain Parkinson's disease consortium. So for today, the science is just it's moving so fast, and I can tell you as a scientist in the Parkinson's field, it's such an amazing time to be in the Parkinson's field, and I'm going to have the pleasure of being able to describe to all of you today how our scientific understanding of Parkinson's has really evolved over the last several years, and why researchers like me are increasingly hopeful with the new science that's emerging. I'm going to focus on three key research areas that I think are really, really being transformative in the field, focusing on alpha-synuclein, mitochondrial function, and inflammation, sort of separately, but really how all of those key research areas are really integrated, and how we're thinking of them now more as a pathway with each other. And then I'm going to be able to identify where I think the current research trends that may shape future treatment strategies for Parkinson's disease. And of course, at the end, I'm very excited to take questions and engage with all of you. So, unfortunately, I really don't need to mention, right, of the big picture about and why this matters. We all know that Parkinson's disease is the most common movement neurodegenerative disorder. It's sort of staggering that the latest numbers are over 11 million cases globally. That's over 1 million in the U.S. Alone. It's sort of remarkable to even think about that. The estimates are by 2050 that were projected to exceed over 25 million cases. And while there certainly, unfortunately, is no cure yet, the progress is accelerating at such a rapid pace that you'll see why I'm so optimistic that all of this research really is leading us to better treatments and towards disease-modifying therapies. And partly due to the Parkinson's Foundation and other Parkinson's disease-related foundations, this is just released. This is sort of hot off the press of that in 2024, Parkinson's disease cost the U.S. $82.2 billion. And I'm sure that's even more in 2025 and 2026. It's hard to even wrap your mind around, right? That economic impact. And while I'm sort of sharing you the numbers of just total, in terms of number of people with Parkinson's disease and the economic impact, I by no means, want to. I think it's really important that this is all of us, right? This is impacting all of us at such a personal level. And so while these numbers are staggering, these are our families, these are our friends, these are people that we really care about. And I've been a scientist in the Parkinson's disease field for a long time, and just want to share that it affects me personally as well. My postdoc mentor, Dr. Tim Greenemeyer, which many of you may know, in Pittsburgh, a few years ago shared in a Science article that I encourage all of you to check out. You can find it. I'm happy to share it sort of afterwards, where he, described being a physician scientist and being diagnosed himself. And so this has really deeply affected me as well. And I'll say that more recently, my mom has been diagnosed with Parkinson's disease. And so I can tell you that I'm have been 100% committed towards moving the field forward towards better treatments and cures. And now it's even more personal for me. And so I can tell you that from my perspective, I'm really optimistic and hopeful, even with all of that I described, that we're really moving in such an exciting direction. So what's driving this progress? So, for you history buffs, there's some really actually interesting ancient scripts talking about Parkinson's disease and sort of the symptomology. And so even over this last several hundred years, and this is by no means exhaustive, really showing from those sort of initial James Parkinson's essays, describing sort of the symptomology and the sort of the movement symptoms of Parkinson's disease to sort of the pathology. And then over time, this has really started to change and evolve and shape how we're thinking about Parkinson's disease. So this is sort of a nice roadmap, I think. Again, not exhaustive of where we've kind of traveled since those initial essay, from James Parkinson's, where in the mid 20th century, really beginning to understand the pathology around Parkinson's disease, and that it's this dopamine loss in the substantia nigra that then really led to that development of levodopa therapy in 1961 as the first symptomatic treatment against Parkinson's disease. And even honestly, from the 60s until maybe about even 20 years ago, really that focus on motor symptoms and symptomatic treatment. But that's begun to change, even since I've started to train in the Parkinson's disease field, where certainly, of course, there's the motor symptoms of bradykinesia and increased risk of falling and others associated. But even the refinement of those motor symptoms and how it might affect one's speech or difficulty in swallowing. But even beyond the motor symptoms is really beginning to appreciate the non-motor features of Parkinson's disease, and that this really extends and goes beyond into sort of this multi-system involvement. And so certainly there's, can be cognitive effects, GI symptoms, urinary problems, sleep disorders, and many others that are sort of not mentioned here, and that we're really thinking of this now as sort of a whole body and multi-system disease, as opposed to just sort of this motor symptomology. And beyond that, and this has really happened in the last 20 or so years, is that due to sort of these, what we call these molecular and genetic insights into Parkinson's disease. And here I have sort of a map of some of the most well-known and now studied genetic risk factors for Parkinson's disease, has really been a game changer for us in understanding what might be causing sort of the risk and the development of Parkinson's disease, and maybe even the progression. And so here on the sort of the lower frequency end, so these are rarer genetic risk factors for Parkinson's disease, like LRRK2 or SNCA, which is synuclein, or even pink and Parkin. So while these are rare, these are really taught us a lot about sort of those molecular changes and insights into sort of pathway dysfunction that is underlying Parkinson's disease. And on the sort of the more high frequency here is over 90 genes now have been associated with sort of a risk at the what we call loci or genetic loci in the development of Parkinson's disease. And so this has really opened our eyes into not just the motor and now non-motor features, but sort of those underlying cellular pathways that may be dysfunctional, which is so important because that gives us really a window into understanding for how we might be able to best treat Parkinson's disease. And so I was sort of mentioning those cellular pathways and what we call sort of cellular pathogenesis. And so that's sort of the underlying pathology that may be driving sort of what we're seeing as those clinical manifestations of the motor and the non-motor symptoms. And so we're going to focus on proteinopathy or that synuclein, mitochondrial dysfunction, and neuroinflammation. And it's really, each of these are critical components to what we call the hallmarks of neurodegenerative diseases and Parkinson's disease. And, I did want to mention there are other cellular pathogenic pathways here in terms of synaptic and neuronal networks, cytoskeletal abnormalities, DNA and RNA defects that also play important roles towards ultimately driving sort of this neuronal cell death and pathogenesis that we see in Parkinson's disease, and where I think our field is really evolving and changing is thinking about how not any one of these separate pathways may be influencing the biology underlying Parkinson's disease, but how they're now all sort of interconnected and sort of what might come first. And then on the other hand, a consequence of many of these other pathways, that sort of more holistic understanding all of these hallmarks has really been breakthroughs in understanding again how to treat Parkinson's disease. But there's, I can't even mention all of the amazing other advances that have been occurring in the field that has really been driving a lot of the progress sort of at light speed. Certainly advances in imaging, so PET ligands, high resolution MRI, other diffusion imaging has given us new insights into imaging into the brain, not just in animal models of Parkinson's disease, but in humans as well, so that we can really begin to understand some of the inflammation that's occurring in the brain, but also some of those protein aggregates such as synuclein. I'm sure and I'm excited to talk about sort of the discoveries of the biomarkers in the field. Certainly the alpha-synuclein seeding assays are also known as RT-QuIC. Other fluid biomarkers such as neurofilament light or NFL, and other inflammatory markers that really begin to, at a molecular level, be able to define that cellular pathway in people with Parkinson's disease. Molecular tools, so single RNA-seq, CRISPR screens, cryo-EM, structures of these synuclein fibrils and other conformations. Improved models. These are all, again, not perfect models, but being able to have sort of induced pluripotent stem cells derived from either people with Parkinson's disease or people without Parkinson's disease, and neurons, microglia, even organoids, little brains in dishes, and even refined animal models gives us better tools to be able to test some of our hypotheses for that cellular dysfunction. It's just sort of a 10,000-foot view too, I think, and this is certainly hands off to the Parkinson's Foundation in terms of really encouraging and fostering global collaboration, not just within the US, but all around the world. Certainly PD-Gene and other efforts that are going around, internationally, so that we as a community can get a better understanding in all different cultures how Parkinson's disease manifests and what might be the sort of genetic drivers. And certainly, thanks to you, probably many on the call here, I certainly encourage you that we can't make sort of some of these discoveries without patient population and research. And it's fundamental for our progress forward. And that's really, and many, many others that I don't have mentioned here in terms of driving progress in the field. And so this has really led to the translational momentum and the clinical trial landscape. I have here the citation of this great sort of summary of all of the clinical trials that are in the pipeline currently. The last one here was 2024. And hopefully what you can appreciate here is that there's a ton in the clinical trial pipeline, certainly in that early phase trials and even the later phase trials of phase 2 or 3. Um, what's exciting to me is if I just did it right before I logged on here to see how many clinical trials are, if you go into clinicaltrials.gov and just type in Parkinson's disease, there's over 200 clinical trials currently either actively recruiting or about to recruit. I think this really speaks towards this momentum of being able to test not just symptomatic therapies, which are obviously critical and key, but also this shift towards testing the disease modifying therapies and being able to hopefully halt or stop the progression of Parkinson's disease. And so there's a lot here to be optimistic about. So this has really, I think, shifted our evolution of our understanding of where in the field we're really historically have been thinking about focusing on that dopamine loss, more motor symptoms, and really thinking it through that primary lens. But now we have a much better understanding of how Parkinson's disease is really this whole body, multi-system disease, and that this includes which is obviously very important, the motor, but also these very key non-motor symptoms that really matter to people, and that we now have tools for earlier detection and intervention that we have ever before. And so hopefully you can tell that that there is a lot of hope, which I share generally. And I think this is really due that this happens when people with Parkinson's disease, researchers and clinicians really work together because we really have the same goal. And hopefully, from what I've explained so far and what I'm sort of going to share now with you, that you all will share the hope that I have with me, that I keep with me every day, that we are working closer and closer towards disease-modifying therapies for people with Parkinson's disease. All right, so what's happening in the brain? Certainly we know that loss of dopamine-producing neurons in the substantia nigra in people with Parkinson's disease, but also is this protein buildup of synuclein and other sort of proteinopathies in the substantia nigra and also throughout the brain. And we'll talk a little bit also about the gut. And how this leads to the cellular stress and dysfunction through the mitochondria and immune system, and how these are all interrelated. So I'm going to first start with focusing on alpha-synuclein. So I think what's really important to understand about alpha-synuclein is that while it certainly is causing havoc in Parkinson's disease, alpha-synuclein has a physiological and normal role in the brain. So this is a protein that's found normally in all our cells in our brain, myself included, and has specific roles in our brains that help us. It's a protein that's known to be in the presynaptic part of our neurons that helps us with neurotransmitter release, such as dopamine, as well as what's called sort of vesicular trafficking, so sort of moving things around in our cells or neurons normally, and other pathways that are really important in terms of learning and other pathways in our brain. And this really, synuclein helps us communicate between neurons and between different parts of our brain. However, what we know happens in Parkinson's disease is that synuclein begins to sort of misfold and clump together, and that this starts to cause sort of pathway dysfunction. And its role is not really fully understood in how it does this, but we know certainly that it's a good target for disease. And so, there's been a lot of work focused on sort of the pathological, I guess, role of synuclein in Parkinson's disease. But I think it's really important to mention that because it has this um, normal and physiological role in Parkinson's disease, or in our brains, excuse me, that this sort of makes it tricky in order to target it, right? Because you don't want to affect the normal and physiological role of synuclein. And so there's been a lot of work in trying to figure out how to specifically selectively target sort of the pathological or the misfolded types of synuclein so we can sort of protect the sort of monomeric or physiological role of part of synuclein. And in our dopamine neurons, which have really interesting biology around them, is that there is incredibly long neurons in terms of their terminals. And so from the syncytial nigra, all the sort of axons and dendrites that go all the way into the striatum are really, really, really long and travel really far in our brain. And the synuclein is not just in the cell body of neurons, but also sort of along that track and that axon and dendrites. And so also understanding whether to sort of target, within the cell body and along sort of that, think of it as like train tracks into different parts of the brain has led to some of sort of barriers in trying to figure out how to target synuclein. And so, from that, monomer, this is sort of what we think of as like synuclein going wrong, is that misfolding and what we then call oligomers and fibrils. And these are these higher order structures and have sort of a picture of that represented here that then form these Lewy bodies. Which is sort of the hallmark of Parkinson's disease in the brain. I would say that there's still sort of some controversy of and context dependency of whether we think those Lewy bodies are protective or are actually causing part of the problem in Parkinson's disease. But what's really come to light over, I'd say, the last decade or so is this idea that synuclein actually can propagate between cells, almost like this prion-like seeding. And it's this interaction with lipids, and those are sort of fats and membranes of different organelles that will then lead to this sort of cellular dysfunction, including mitochondrial effects and inflammation. And that's what we're really trying to target in Parkinson's disease. And there's very strong evidence for a genetic and a pathological evidence linking synuclein to Parkinson's disease. We know that point mutations and multiplications in the synuclein gene cause familial Parkinson's disease, and there's a dose-dependent severity here in terms of clinical severity. There are genetic modifiers such as GBA and LAIR2 that actually influence the biology and risk with regards to synuclein. We know it's a central pathological hormone of Parkinson's disease, although I will mention that even beta amyloid and tau have become really significant and important too in terms of these protein aggregates that are co-occurring in the brain. And we know that synuclein pathology is correlated with cognition and linked to disease progression. And all of this sort of taken together has really founded the foundation or rationale for many different approaches for targeting synuclein and the development of really some groundbreaking biomarkers around synuclein pathology that we can then use in clinical trials in clinical practice. Okay, so there's many I think key questions and challenges that are still ongoing in the synuclein field. You perhaps might have seen that there's been proposed this biological definition of neuronal alpha-synuclein disease as defined by the seeing amplification assay. I think there's a lot of controversy around this and what this means, sort of clinically, and sort of using a biological definition instead of a clinical one. We'll see if some of the questions come up around this, and happy to talk about that. It wouldn't have been possible to sort of have that biological definition without the seeding amplification assay. I will say one of the barriers and challenges there of the seeding amplification assay is that right now works really well in the CSF or from a lumbar puncture, but obviously that's very invasive. Currently, there's no blood iteration of the seeding amplification assay, but there's a lot of people working on that to be able to get it to work in the blood. And so I think we're going to see some really exciting developments in the next couple of years about that. Actually, I think one of the most exciting things that's come out in the last really couple of years is that in addition to sort of these oligomers and fibrils that I talked about, these different higher order conformations of synuclein, there are so many different conformation and forms of synuclein, close to 100, and there's probably even more. And so I think the real question is, which are the most toxic? And that's important because which are the best to target? And so that's where I really see part of the synuclein field putting their efforts towards better understanding that. So I kind of focus on the brain, talking about synuclein, but now we know that synuclein is not just found in the brain. Synuclein is also found in our olfactory system as well as the gut. And so there's these really interesting hypotheses around sort of gut first and then into brain in terms of development of Parkinson's disease, or brain first and sort of going the other way to the gut. And so this is sort of exploding research in the field, which is really exciting. And I'm really pleased to share with you, this is our website just went live for our gut-brain Parkinson's consortium, because we think it's really important to be able to really define sort of those GI symptoms in sort of a snapshot and then sort of longitudinally over time. And so you can find out more information about our human study so that we can really begin to answer, I think, some of those questions of the sort of peripheral versus more central nervous system or brain origin. The heterogeneity in that not all Parkinson's disease, I think, is equally driven by alpha-synuclein biology. There's emerging evidence for tau, especially in LRRK2 Parkinson's disease. And so how do we think about sort of targeting that? And I think this is one example of working on a drug, buntanetap, in which targets synuclein, tau, amyloid, and many of these other types of co-pathologies. And so I think this is a really interesting perspective in thinking about how we can really tackle the disease from many multiple angles. Again, towards that optimism, there are many therapeutics that are targeting right now or in development for synuclein in terms of reducing production, aggregation, clearing these aggregates from immunotherapy perspective, blocking the propagation, enhancing the clearance, boosting glucocerebrosidase activity, which is that GBA protein and enzymes that are intimately involved with synuclein clearance and proteostasis enhancers. Those are just the general categories of all the exciting developments that are happening, and just in focusing on synuclein and trying to be able to reduce that pathology burden. So much to be excited about. Okay. And I could talk all day about Tanuka, but we're going to fix it to mitochondria. Again, another love of mine. Certainly that's close to my heart in terms of thinking about mitochondrial function. So what are mitochondria? So I think probably, they're well known for being sort of powerhouses of the cell and sort of produce energy for important brain function. Um, However, what I'm hoping that you all walk away with today is that the mitochondria are so much more than just producing energy for the brain. And so they are these, they're double membrane bound organelles that are found almost in every cell type in your body, and they contain their own DNA. So as opposed to nucleus, where it has chromosomes, the mitochondria actually have their own set of DNA called mitochondrial DNA that encodes many proteins and RNA and all these things, just like the nucleus. And there's a lot of crosstalk actually between mitochondria and nucleus to maintain sort of homeostatic function for the cell. So in addition to obviously the respiration or sort of energy functions of mitochondria, they do so much more. And so this is just a pictorial example of that there's protein import and processing that happens at that double membrane of the mitochondria. And so sort of shuttling these important proteins, and sometimes actually synuclein and tau get inside aberrantly into the mitochondria. There's the biosynthesis of really important iron sulfur clusters and other cofactors that are used for not just function within the mitochondria, but also outside of out of the cell that are critical. Um, the mitochondria not only have the cross-talk to the sort of nucleus that I spoke about, but have all these other organelle contacts, such as the ER and others, that really help us maintain sort of that function and homeostatic function. Um, as opposed to sort of the nucleus, mitochondria can be selectively targeted and purged from the cell when they become sort of dysfunctional, sort of wear out during a normal aging process, during a process called mitophagy. And we think that this is actually dysfunctional in Parkinson's disease. And so it's sort of this accumulation of dysfunctional mitochondria that build up. But beyond that, sort of signaling and all these readout processes and the metabolism of amino acid, lipids, and nucleotides, and the transport of these metabolites, in addition to the energy metabolism, and all of this think of the mitochondria managing all of these processes and really important functions of the cell, that when things start to go wrong, actually will send sort of a signal to the rest of the cell or neuron in this case for cell death. And so mitochondria are key in terms of that signaling of whether or not that neuron will survive or not. And so there's many consequences to mitochondrial dysfunction, in addition to obviously this energetic failure, but an increase of this oxidative stress or reactive oxygen species, impaired mitophagy as a consequence or cause of dysfunctional mitochondria. Where we focus a lot of our efforts in terms of damage to that DNA, that can either lead to mitochondrial DNA damage or mutations that we know definitely happens in Parkinson's disease and ultimately can drive cell death. And we know that neurons are especially vulnerable to this mitochondrial dysfunction. And so this is a really very exciting area of research, not just in my lab, but many others around the world. There's a strong rationale that of many decades of research that's been done, both sort of in the preclinical space as well in humans living with Parkinson's disease, that really sort of substantiate this idea that mitochondrial dysfunction is playing a key role in the pathogenesis of Parkinson's disease. We know from toxins such as paraquat, and PTP that when administered to animals reproduce not perfectly, but reproduce many of the features that we know happens in human Parkinson's disease. We know that this is due to complex 1 deficit, so it's sort of that first initial part of the respiratory chain that helps us produce our energy in the mitochondria, that there's this sort of specific complex 1 defect that we can reproduce with many of these toxins, but also we know from human Parkinson's disease and tissues that there's this sort of complex 1 defect in not just in the brain, but also systemically in muscle and blood and other tissues. We know that mutations of select genes cause Parkinson's disease. And so some examples of that are DJ1, PINK1, Parkin, LRRK2. More recently, we know that even POLG, that mutations in POLG will lead to nigrostriatal degeneration in carriers of mutations with POLG and sort of look Parkinsonian. Many data that I can't even describe here is that we certainly know that mitochondrial pathology has been observed both in the brain and the periphery in many of these monogenic forms of Parkinson's disease, as well as idiopathic forms of Parkinson's disease. And why this really matters is that our brain cells really need this high energy. Our brain cells are one of the highest consumers of this high energy demand, and as that dysfunction starts to occur, this contributes to risk and maybe even disease progression for Parkinson's disease. So I just want to briefly comment that while we've certainly known that mitochondria dysfunction is such a key part of the pathogenic cascade in Parkinson's disease, that what's been sort of a challenge in the field is the development of a biomarker to be able to use this for clinical trials and clinical practice. And so we've spent a lot of efforts towards developing a biomarker around mitochondrial dysfunction. And there's many different types of biomarkers. And the one that we've really concentrated on is the one to be able to patient stratify. And what that means is to be able to identify people with Parkinson's disease with that particular cellular dysfunction so that we can then sort of match that person with Parkinson's disease to the right therapeutic with that biomarker. And so of course, I'm always happy to talk about this in great detail, but we've been able to really develop our mitochondrial DNA damage as a blood-based biomarker of early Parkinson's disease. And this was enabled through technology called MitoDNA-DX. This is published, and I'm happy to sort of geek out in the technology here. It's in the question and answer session, or later you can email me. I'm always happy to geek out about this. In that we're essentially able to measure whether mitochondrial DNA damage is out of homeostasis. And what that means is if there's an increased DNA damage or sort of a decrease that might be due to DNA repair or mitophagy or others. Why we've been able to move this along the translational pathway is that this is a PCR-based assay. We can do this in sort of a high throughput manner, and we can go from sample to answer in a day from a blood sample. And so we're really excited about moving this. And so this is been published. You can find some commentaries about our work, about our blood test. I'm really excited to share that we are moving this along so that we can be able to use this in clinical trials and hopefully ultimately into clinical practice, so that we really can start to use this as a way towards precision medicine. And so I think, I didn't mention this in the beginning part of sort of how we're, evolving in how we're thinking about Parkinson's disease. But part of that is really this precision, or what might, you might know as a sort of personalized medicine approach. This was really coming, I think, to Parkinson's disease clinical trials. So, our assay, or thinking about mitochondrial dysfunction, is just one example of that. But this can be sort of applied to any, pathways of lysosomes, immune system, synuclein, right? Where we're really being able to, at a molecular level, be able to identify really that underlying pathology, so that we can identify, as we say, patient stratify, and find that person with Parkinson's disease with, let's say, that mitochondrial dysfunction, so that we can then best match them to a mitochondrial therapeutic, for example. And equally as important is stratify out those that would not benefit from these drugs. And so I can't tell you how exciting it is that we are really moving in this direction in Parkinson's disease. And so this sort of precision neurology approach, this has been taken in cancer, right, for decades and decades, but it's really coming to neurology and Parkinson's disease in terms of the biomarkers, as well as therapeutics, and really evolving our way in which we can design clinical trials and hopefully be able to get approvals and an FDA win. So I think one of the really open questions in sort of the mitochondrial function space is, really, mitochondrial dysfunction, is it really the driver? Is it the cause of many of those downstream cellular dysfunction? Or is it secondary? Is it a consequence from synuclein and immune disturbances? And so really distinguishing that causality versus consequence. And that might be really different for different people, which makes it a challenge, but certainly one that I think that we really need to tackle. An interesting, I think, development in general in the mitochondrial field is this idea of mitochondrial donations, where you can take a healthy mitochondria and actually donate and transfer them from cell to cell. And so I really have my eye on those developments, and if they should prove successful, how we might be able to apply those in Parkinson's disease. But there's a challenge in these beautiful neurons that we have in our brain, especially these dopamine neurons. As I said, those are really long axons, right? And so how do we deliver some of these therapeutics to certainly mitochondria in the sort of the cell body versus these long axons or the dendrites that are really far from the cell body? Because the mitochondria actually, in terms of their dysfunction, differ whether they're sort of a really far away from that cell body or not. Now, hopefully you're taking away that mitochondria are much more than these sort of sources of energetic function. And so how do we modulate that mitochondrial function safely, where we can rescue maybe some of those defects without harming some of the other functions of mitochondria? As I mentioned, sort of that patient level heterogeneity, you're going to see that sort of in all of these sort of key challenges. And then I think, not everybody is manifesting the same sort of pathology, even though sort of the clinical manifestations may be the same. And so how do we account for that? And I think part of that is through biomarkers. Certainly I'm excited about all of the developments in terms of mitochondrial therapies for Parkinson's disease. And these include mitophagy enhancers, metabolic enhancers, some of these through the NAD pathways that some of you may be familiar about, and gene therapy approaches to sort of rescue mitochondrial function. Okay, so inflammation. So now we've walked through sort of the mitochondria and synuclein, but equally as important is the inflammatory cascades. So just like you might think about inflammation throughout the rest of your body, it's your body's response to sort of harm, and this is where your brain cells, such as microglia or astrocytes, become active. And then they release these chemicals in trying to protect or sort of clean up that damage that might be occurring in your brain. For the most part, the response is short and very well controlled, and it's helpful. However, when the inflammation response becomes chronic or too strong, this can actually damage a healthy brain and make some problems worse. And so we think that some of that might be happening in Parkinson's disease. And so, one of the brain cells, microglia, you can think of as sort of the cleanup crew. They sense, the trouble, and most of the time, they're helping with sort of that cleanup. But when they become really activated, which again, we have evidence in Parkinson's disease that they do this, that it starts to harm those connections and signaling. Now, what's interesting is that, synuclein aggregates, as well as dying neurons, can actually trigger microglia to switch on. And this releases those cytokines, inflammatory and other toxic substances, and which will then increase further that inflammation, right? And so really thinking about that there's this vicious cycle between, the inflammation trying to help, but then it gets activated and it can't sort of turn off. And so, during, normal healthy aging, what happens is that there's this decline in sort of immune cell function, with also sort of this immunosenescence, and actually loss of some of these functions of the immune system. And we think that's sort of enhanced in Parkinson's disease. So there's sort of that aging, normal aging that's happening, in addition to certain gene risk factors and environmental exposures, that's all sort of conferring this risk and maybe contributing to sort of that vicious cycle with our immune system, but also synuclein and mitochondrial dysfunction. And as part of this, our blood-brain barrier can become leaky. And what that means is that the immune system that normally is in the periphery can now get into our brain. And this is really important because now sort of there's this two-way highway that doesn't exist physiologically, and sort of contributing to sort of that immune activation. This is really significant as now we know that that immune activation is not just happening in the brain, but it's also happening in our gut. And that also may be happening through sort of our vagus nerve, sort of in two-way traffic. And why this really matters is that now this is connecting multiple disease pathways. And I said this is really integrated now. None of these pathways are happening sort of in silos or in isolation. And that this actually may accelerate disease progression, but also gives us opportunity with our better understanding of this as a potential for new treatment targets, that we can actually target all of these simultaneously. So there's a lot of research momentum, certainly in inflammation, with many strategies to reduce sort of those immune responses, to sort of dampen them, so that hopefully we can develop some of these neuroprotective strategies, and really also take advantage of that better understanding of that role of the gut-brain connection. And this has been a development of, drug development focus from many different angles. Some of the, I think, key research questions that stand out, at least to me, is that, is inflammation friend, foe, or both? I think the answer is probably all of them, but it's really going to matter in terms of stage of disease, and it's going to be really context and cell dependent, of really understanding and when we want to harness the immune system and what its job is normally doing, but then also to be able to dampen it when we need to, that it might be causing that activation that we want to turn off. What I find particularly exciting that's happening in the last few years in sort of the inflammation space in Parkinson's disease is that we can actually measure some of that immune activation in the brain with PET imaging, what's called TSPO. And so in addition to some of the very other exciting imaging in the brain that we can use, we can now do this and understand better who has this sort of activation and inflammation, and who may not. Um, where it starts to really tie together is this idea of immune exhaustion and how this may be caused by metabolism and mitochondrial dysfunction. And hopefully, what you're gathering by this point is that how these are all interconnected. And so, synuclein can be driving also that immune exhaustion and mitochondrial dysfunction, and vice versa, and how these are all intertwined together. Again, that heterogeneity, where thinking about these immune signatures that really vary across people with Parkinson's disease and at different stages, and that gives us a real opportunity to target in terms of dampening or supporting the immune responses. We are not at a loss. There's a plethora of different therapies that are in the pipeline targeting inflammation. Obviously, that anti-inflammatory perspective, but also specifically microglia and innate immune modulators, the adaptive immune strategies specifically, and how that might be different from the innate strategies. What if we just target the peripheral immune systems and taking advantage of what we now know in terms of dysbiosis of the microbiome, also gut-targeted therapies, and how that might actually help in the brain without it even directly in the brain, and even vagus nerve stimulation, and how that might improve brain function. So bringing it all together, of all these exciting developments that are happening across synuclein, mitochondrial function, and the immune system, is that there's so much crosstalk between synuclein, mitochondria, and the immune system. And it's not just in the brain. This is a whole system and multi-system disorder that we now, which is such a better understanding of this, that there's this crosstalk between what may be happening at the immune cell system as a result of that mitochondrial dysfunction, and synuclein triggers, or vice versa. You'll see that these arrows are sort of going in both directions, right? And really understanding what might be sort of initially the trigger versus what might be sort of perpetuating that vicious cycle between all of these different pathways. And that this is not just happening in neurons, it's happening in the immune system, in our brain and peripherally, as well as in our guts and other tissue types throughout our bodies, and really understanding how that is now contributing to not just the motor dysfunction, but many of the non-motor features of Parkinson's disease, which are so important. So we're not really thinking about these laterally anymore, right? It's more of these really how synuclein inflammation and mitochondrial function are all sort of intertwined, and that these are interconnected, and no longer thinking of these as isolated issues. So I will continue to maintain why I am so hopeful, and that we're really moving beyond these dopamine-only therapies. And this has only been possible with a really stronger understanding of disease biology. And this is, due to early detection possibilities with a seeding assay, better tools and technology for imaging, and all the other things that I mentioned, where we're able to now really be able to target disease progression. I feel really optimistic of these disease-modifying therapies that are in the pipeline, and many growing pipelines for the clinical trials, and that we can move not just towards these away from just dopamine-only therapies and really disease-modifying, but really that personalized precision medicine approach to be able to identify the right target and therapy for the right patient in Parkinson's disease. So what this means for all of you that are here today is that while symptom-only therapy is key and is still in development for better treatments for symptoms, but we're really light years going in for development for slowing progression. And that it's this shift towards disease-modifying therapies in our clinical trial pipeline. And that the research is ongoing, evolving, and so fast. And it's only because of the participation as well for all of you in the research that's helping us move at such a fast pace. And it's important because then we can all make informed decisions about our health, and that really empowers us moving forward. So what's ahead? Certainly biomarker development in Alzheimer's disease and Parkinson's disease is allowing for better and earlier diagnosis, which is really feeding this ongoing clinical trial space. But also, which is an area that we're starting to get into as well, is really thinking about these combination therapies. Um, we know now all of these pathways, right, are intertwined and sort of feeding each other. And so can we use combination therapies to really tackle these multiple pathways so that we can have a better outcome for these disease-modifying therapies? And I'm really excited about the development of those. So the key takeaways for today is that understanding Parkinson's is evolving rapidly, and I'm sure it's going to be very different even 10 years from now. There's three major drivers: alpha-synuclein pathology, mitochondrial dysfunction, and inflammation cascades. And we're really, moving towards not just treating the disease, but really being able to tackle the underlying pathology to develop these disease-modifying therapies. So thank you so much for listening and your attention, and excited to sort of engage with all of you during this question and answer period of time. I thought it was also important to, show some of the faces that are doing the research, that have such an amazing crew of dedicated researchers that are really dedicated and committed towards making a difference for people with Parkinson's disease. And, want to thank them and certainly all of our funders and collaborators that really help us move and make this research move forward. So I'll stop there. Thank you.
Thank you very much, Dr. Saunders, for that excellent presentation. And so before we jump into questions, I just remind our audience that our team is organizing questions as they come in, but understand that we can really only address, because we've got an overwhelming number of questions, only address the ones that are really specific to today's subject. And those will be addressed first. If we have time, we can get to some of the others, but that we look to answer just about every question that comes in. So either here or through my colleagues who are eminently capable on the helpline as part of that process. So that really interesting presentation. It's just I think it's really great to hear as we begin to think about the trials that are coming in Bay. They're coming to the fore. What three areas are, and I like the idea of your focus on biomarkers and gut brain, and we've got questions that are ranging the whole course of what you covered. And it's amazing these questions that come in as part of the process. I think one of the interesting questions I'd like to start off with is from one of our listeners, Ronnie. Just talks about how no two people with Parkinson's have the same symptoms. So how does that impact your research? You've mentioned a time and time again the heterogeneity of this disease, and how do you account for that when you begin to take a look at this? And then it's I think also again you kind of touched on it maybe a little bit with your biomarker approach, maybe mitochondria. I mean, do you think there might be a biological basis to this heterogeneity?
Yeah, thank you. Thank you for that question, Ronnie. I will say that this idea sort of heterogeneity and that person with Parkinson's disease, their journey, right, looks so different from one another, right? And that sort of experience, right, it's just so different, right? And that really struck me as a postdoc, actually, where I thought it was really important to meet and talk with people with Parkinson's disease to really understand, right, from their perspective, right, of what's happening. And what really struck me, and I think that that's really changed the course of how I study the this in the lab, is that I think you can't model a lot of that. And that this is a real heterogeneity, right? That people are experiencing from a biological perspective as well as a symptom perspective. And so one approach that we've taken is that we start with the patient first, and then we go back to the lab to model it. So we do sort of human studies, and sometimes that's taking patient material from a wide range of people to best understand what might be happening across sort of that patient experience, but we can see that sort of molecularly, and then take that understanding and model that particular aspect in the lab because our models are just not doing that justice. And then from that, from what we get sort of from those sort of modeling in cells or neurons or animals, and then go back and see if it actually works in our human cells. Because to get sort of at your question is, I think that's been a lot of the barrier to the progress in being able to really be able to nail down and understand that heterogeneity that we know is happening in human Parkinson's disease.
Yeah, so that's really interesting, answer, and just makes me think. And what one of our listeners also had the foresight to ask this question as well. Charles, it's so I mean, I appreciate the effort to try to model what you're seeing in people and animal models, um, and as a researcher, I maybe this is an unfair question to ask of you because I kind of know the bind you're in, but is it is it the reason you think our clinical trials have failed to date? Is it because of our lack of ability to model adequately? Or is it something else that you're working on too, which is trying to find a way with which to measure PD, the biomarker approach? What do you think is maybe behind some of the struggles the community has faced to really kind of put a stop to this disease?
Yeah, no, that I mean that's such an insightful question, and one that we think a lot about. Unfortunately, right? Like we all want wins, right? Of being able to get these approvals, and why are these clinical trials failing, right? Why are they not reaching endpoint? Um, I think part of it is our inability to model the complexity of the pathways and cellular dysfunction that we know is happening in human Parkinson's disease that are their models are just have been inadequate in that way. So that's one piece of it, and so why I kind of think of it a little bit. We distill a little bit. We know we're just studying that one angle, um, but also I think, is sort of alluding to, now that we have a better understanding of the complexity and how these pathways are sort of interrelated, that we've been doing one at a time.
Yeah.
And what if we took more of a multi-pronged approach and targeted multiple of these pathways at once? And I think that's also part of why we've not seen successes. And that we really need to tackle the immune system, synuclein, mitochondrial and others, right? In order to really move the needle. And I think that's also, I mean, there's a lot of reasons, but I think that those are two really key reasons as to why I think those have been such barriers in clinical trials.
Yeah. Okay. That's a very appreciate that answer because it's one we clearly don't know the answer to, right? Because if we did, then we could solve it for it, and then we'd be that much closer to a cure for Parkinson's. A lot of questions come in about aggregation of alpha-synuclein, and it's interesting how you also started at the very beginning. And I do think it's controversial, this idea of neuronal synuclein disorder, not in the sense that it's bad to try to define Parkinson's disease as a biological disease. Right now, it's a clinical presentation, and I think everyone in the audience knows firsthand the implications of that, which means it's hard to diagnose. your diagnosis is uncertain, and it can be a long time to do it. And if there was a biological basis of the disease, like with diabetes, oh, your A1C level is above the norm. You need to really target your insulin level to this. It would be entirely different for us as a community to be able to tackle this disease. So a little bit more. So getting to the point about aggregation with alpha-synuclein, what are your thoughts then about is it a are we looking at the bystander of what's contributing Parkinson's? Because you've talked about mitochondria, you've talked about inflammation, and these things could lead to the proteostatic dyshomeostasis, where you have problems with proteins inside cells. Sorry, use the right words for my audience, and then is that leading to this? And then we now see this cyclic effect where in essence, you've got something like staying in the normal, something pushes it out of whack, and now it just becomes a spiraling cycle. So lots for you to go on, Dr. Sanders. So maybe just narrow it down. Let's talk a little bit more about aggregation and alpha-synuclein, maybe as a start.
I certainly acknowledge that is very a tough question, right? Very controversial. So I will try to answer that. You know, and again, I think it's useful to have sort of at least definitions that we can then sort of use and build upon, much like the Alzheimer's disease field, right, in terms of these biomarkers and staging a biological definition. I will say where I struggle is that there are, for example, in the LRRK2 field, we know that patients with the LRRK2 mutation don't have synuclein aggregation, but they have Parkinson's disease, and they're negative, right, for this. And so, in one answer to your question, I will say that we know you can get to Parkinson's disease without maybe that trigger of the synuclein aggregation. Now, it might be in the there might be Laurie neurite pathology. There's been a couple papers to suggest, right, that maybe you don't have the Lewy bodies, but maybe you have sort of these Lewy neurites, right? That's also controversial. So there might be, but it's not this sort of full-blown Lewy body, disease that we know, right, that defines. So, I think there are multiple ways in which you could get, and it's not always through synuclein. That being said, we know that synuclein pathology happens really early. It could be in the gut, decades before somebody might, manifest clinical symptoms. And so certainly over time that's going to drive many of the clinical symptoms that we might see, and certainly can drive disease, right? So I'm not sure that that's an answer, right? Because there's just these examples of different ways. And I think that's, understanding that, maybe you get to the same point, right? Eventually,
Yeah.
it may not matter, right? From that perspective of sort of a symptom but it's going to matter for disease modifying therapies of what those initial triggers are. And I think that's where we need to define it, and is really critical for some people, right, that synuclein pathology is driving the mitochondrial dysfunction, is driving the immune system dysfunction, and that's when we need to intervene and how. But it's not going to be the same for each patient.
Yeah, for sure. Pesticides are something that are topic du jour. Lots of questions have come in about that, and I think about that as we run a large genetic study with PD generation. People come back negative, yet they have a family history of Parkinson's. And I think one of the biggest things that we haven't really talked about and beginning to explore as an organization at this foundation is what is the environmental influence for Parkinson's. Do you see, in your work, again, because what role, your thoughts just broadly about what pesticides might be doing here, what kind of toxins might be playing a role within Parkinson's?
Yeah, absolutely. Of course, I will say, from doing my postdoc in Tim Greenemeyer's lab, I certainly have thought about sort of, since his rotenone discovery and with Carly Tanner, showing risk, obviously, for developing Parkinson's disease. At least the way I think about it is that at least what we've been able to identify in the field is, increasing one's risk for developing Parkinson's disease. They have a common sort of denominator in the way that they're working in the cell, and that's been through mitochondria. And so I think at least where I would see sort of putting the efforts is that understanding that pathway dysfunction and then how that's sort of generating disease, as opposed to the specific exposures.
Yeah.
Um, uh, that's at least where I would put my efforts because there's a lot of even just plant derivatives that mimic these complex one right disruptors, and I think that's what it's really telling us that they're working through. And maybe there's some other types of exposures that might be working through different mechanisms that would be of interest, because then we know at least where it is that we could start to block or be able to sort of understand that dysfunction, as
Yeah.
opposed to the specific exposure per se, if that makes sense.
It does. And I think what you're alluding to is really um, it's the pathway, and we have different drivers of it, perhaps the same pathway here you're talking mitochondrial dysfunction. So if one can figure out how to solve that problem, you might be able to then prevent PD from a toxin exposure, because if it's driving that same pathway, then a medication in theory could be a benefit for everyone, whether it's a genetically driven form or not, which I think is really exciting. And this is where I think the field, like your work, is helping to advance, to find these commonalities from different aspects that could be driving the disease. I know are at time right now, so I want Dr. Sanders, I want to thank you very much again for your time today and really presenting what I thought was a really interesting topic, led to a lot of questions. I apologize again, we haven't been able to get to them all as part of that process. But I think this is something that's um, will be for us to food for thought as we go on. I know we have a discussion around pesticides later this year in November, and coming up in May. We have some really interesting things about AI involved in Parkinson's, and I'd love to think about how that can happen today. So I want to thank everyone for their time, and just a reminder that um, to continue on with our program and to follow on with what we have coming up. We've got one more episode in May, as I said, before we take a pause during the summer and continue again in the fall. If you have questions, sorry, I'm a little out of order with our slides. If you have questions, feel free to reach our helpline, 1-800-4PD-INFO. My colleagues there are phenomenal and can really answer a wide variety of questions, either from today or just around Parkinson's disease in general. So really encourage you to do that. And I would like to just at the end thank again Genentech for their support for this particular expert briefing. It was fantastic. And also like to thank the Light of Day Foundation for supporting the Health at Home series. They are a great a supporter of education and outreach for people with Parkinson's disease. So thank you very much for their generosity for doing this. As we end the webinar, we're a Zoom world, and so what will happen, the screen will go black, but what should happen is a questionnaire pops up in your browser. Please feel free to fill that out. We use that feedback. It's really important to us to understand how we can always improve our presentations, share that with our speaker, and be able to ensure that we're there and meeting your needs as a Parkinson's community. So until we speak again in May, take care.